Good afternoon, everyone. My name is Josh, and today we'll be covering cellular energetics, also known as cell metabolism. When it comes to cell biology, there are two types of reactions we need to consider. One of them is called the exergonic reaction, and the other one is the endergonic reaction. In exergonic reactions, these are the type of reactions in which the reactants have high amounts of free Gibbs energy, and the product has low amounts of free Gibbs energy. Essentially, this is the type of reaction in which there is heat and light that's being released, and the end product is a product that has very, very low Gibbs energy. Alternatively, you have the endergonic reactions, which are types of reactions that require an input of energy such that the final product has high amounts of energy. To give you an example of each of these, you could think of the exergonic reaction as the one in which you might have, for example, sodium chloride, so NaCl. So sodium chloride, so NaCl, which is also known as table salt. In the process of making this, you combine sodium metal plus chlorine gas. Each of these individuals are very reactive. So sodium metal will react in water and explode. Chlorine gas is very toxic. If it goes into our lungs, it will potentially harm us and potentially kill us. But when you have these two elements combined, in the combination of these two, you actually produce light and heat energy. And the final outcome is sodium chloride, which is also known as table salt. Table salt is harmless, doesn't harm us in any way. We could eat it, does not kill us. Then you have the endergonic reaction. Endergonic reaction is very interesting in the sense that you could think of it as a type of reaction in which you are increasing the amount of energy that's contained within simple reactants. For example, in the process of photosynthesis, in which we use CO2 plus H2O, which each of these, carbon dioxide and water, are very stable. They don't react with anything. Water and carbon dioxide don't blow up into flames when they come into contact with oxygen. But with the application of sunlight, these products end up making glucose, which has a lot of chemical energy in it in its bonds, and also oxygen gas. And if you put oxygen in a balloon and light it on with the fire with the match, it will explode. So essentially, you could see that you need to input large amounts of energy such that the final product has more energy at the end than the products do. The same thing for this case, glucose has a lot more energy, oxygen has much more energy than CO2 and hydrogen and water on their own. Now, how do chemical reactions take place? So chemical reactions move forward as a result of activation energy, which means that this is the energy that's necessary to push the reaction forward. All chemical reactions, regardless if they're endergonic or exergonic, require some form of activation energy. On this graph, you'll see that you have your y-axis that denotes energy, and the reaction coordinates, which mean the rate at which the reaction is moving forward. So, and then in the purple curve, you'll see that you'll see a curve that demonstrates. This curve represents the amount of energy necessary to overcome that barrier, that energy barrier. So you could think of it as pushing a boulder up a hill. So you have to put a lot of energy and effort to push the boulder up the hill. And once you reach the top of the hill and apply slightly more energy, the boulder will roll down the other side without you having to push it. So this just means that the energy that's necessary to push the chemical reaction forward. Just to give you some detail, the top of that curve is also known as the transition state, which means that this is the point at which the chemical will not go back, but rather push itself forward and will not stop. Now, you'll see also the green curve at the bottom. That green curve, that is a curve that represents the same chemical reaction, but in this case, there is a catalyst present. Catalysts play a role in lowering the activation energy, meaning that there's less effort necessary to push the chemical reaction forward. And so therefore, you could see that the amount of effort is a lot lower, but regardless if you have a catalyst or not, the product will stay the same at the end. But what are catalysts? What type of catalysts do we use in cell biology? In cell biology, we use something called enzymes. So enzymes are, by this definition, enzymes are biological catalysts that speed up chemical reactions within the cell. In cell biology, or in biology in general, there are a variety of different enzymes. Enzymes are essentially proteins that produce a variety of functions. And top ones that I give examples for this, for example, are amylase, which are found in our saliva that break down starch. You have protease, which are found in our stomach to break down proteins, so from the food that we eat. And you also have lipase, which is found in our gallbladder and our lipids to break down fats, lipids, oils, etc. And then there are other types such as RNases that break down RNA, DNases that break down DNA, collagenase that breaks down collagen, etc. So depending, and then how do these enzymes function, and why are there such variety of different functions of different enzymes? And this is because enzymes, being that they're proteins, are composed of amino acids, and the structure and the unit organization of the amino acids give rise to the function of the enzyme. So essentially, the structure of the protein dictates its function. Now, here's a very good representation, just a general representation of how enzymes technically look and function. So you have a substrate, which are the reactants that you're interested in to convert into a product. And you have this bond. So maybe over time, over millions of years, the bonds will break. So the reaction might move forward in the future by millions of years of light exposure, what have you. But with the assistance of enzymes that help speed up these chemical reactions, especially within our cells, you're able to achieve this much faster. So you have these specific sites on the enzyme known as active sites in which the substrate will come into contact. The enzyme and the substrate form together to create something called an enzyme substrate complex. And then at the end, the substrate is converted into a product, and the enzyme releases the product and is ready to perform the chemical reaction again. So enzymes play a part in pushing for the chemical reaction, but it does not alter the final product. Okay. Now, in biology, there is something known as the induced fit model. This is mentioned in the AP Biology Review textbook, and which dictates that enzymes are able to change their shape or their conformation such that they're able to fit around the substrate so as to induce the chemical reaction. So as you can see here, it seems as though this part of the substrate fits here very well, but this one not so much. This induced fit model theorizes that the enzyme can change its shape, allowing the substrate to actually fit in. And then the chemical reaction takes place, create an enzyme products complex. We talked about the enzyme substrate complex. So upon the initial contact creates the enzyme substrate complex, then after which it produces the enzyme product complex. And at the end, you have the products detached from the enzyme. So again, the enzymes help play a role in catalyzing the reaction forward, but does not interfere in the purity of the final product. Now, however, some enzymes are interesting in the fact that they are sometimes not activated. They need to be activated through the assistance of cofactors or coenzyme. So cofactors or coenzymes are things that attach to the enzyme to activate them. Typically, cofactors are inorganic. So these are more like metal ions, inorganic. And then you have organic, and these are typically vitamins. So essentially, the enzyme does not function on its own. Some of them don't function on their own. You need one of these cofactors or coenzyme to attach to them. The enzyme activates and is able to perform the chemical reaction necessary to convert the substrate into a product. Essentially, this is the reasons why doctors and nutritionists recommend us to have a well-balanced diet such that we could get all of our vitamins, all of our minerals, and if not, take a multi-supplement that contain all these vitamins necessary to help us achieve the homeostasis, which is biochemical equilibrium in our bodies. Now, there are factors that affect rates, the rates of reactions or reaction rates. One of them are temperature, and the other one is pH. So temperature is interesting. So in chemistry, you studied that increasing temperature increases the kinetic energy, meaning that the interaction between the substrate and the enzyme is faster. And then you have pH, which indicates if something is acidic or basic. So in other words, the amount of protons present inside the solution, and temperature, again, is just temperature. It's Celsius. In this case, for science, we talk about Celsius, degrees Celsius. Now, in enzymes, so when talking about enzymes, certain enzymes, and we have a variety of different enzymes in our bodies. We have some of them in, but typically for cell biology, the optimum temperature is about between 37 degrees Celsius to 40 degrees Celsius. Our body temperature is average on 37 degrees. When we have fevers, we reach sort of high fevers, so about 40 degrees Celsius. But past that, the enzyme will begin to degrade. So it's the same thing as cooking an egg. The egg, when you crack an egg over the frying pan, it's watery, quickly becomes somewhat of a solid. And then if you keep it on longer, it will burn. So it's no longer edible. So again, similar principles apply, that enzymes have optimal ranges of temperature, but beyond that, the enzyme will denature, meaning that it will change conformation and the shape is no longer the ideal. So again, we talked about how the shape of the enzyme dictates its function. So if you change the shape of the enzyme as a result of high heat, it can no longer function as intended. Now you have pHs. In our bodies, we have the pH. The typical physiological pH is about 7.4, but in different parts of our bodies, we have different pHs. In our stomach has a pH of about 2, and our large intestines have a pH of about 8. So essentially, the enzymes that are found in my stomach function optimally at that pH. But if I put them in a pH that's higher, the enzymes in my stomach will not function. And same vice versa. The enzymes in my large intestine cannot function in my stomach because the pH is so different that it doesn't allow the enzyme to function at its optimum form or optimum rate. Now, we talked about briefly that all chemical reactions need energy. We talked about activation energy, or how the curve has to be brought down by the application of a catalyst. But essentially, reaction coupling and ATP, essentially this means that there is energy that's being inputted to create ATP, which means that you need energy to convert ADP and the phosphate group into ATP. And when you break the bonds of adenosine triphosphate, also known as ATP, energy is being released, and that energy is being used for other things as well. In cell biology, typically the majority of ATP comes from cellular respiration. So we'll talk about that in a little bit. But a preview of this is glycolysis, oxidative phosphorylation, chemoosmosis, and in terms of for plants, which are autotrophs, the energy source is glucose. So they use photon energy, which is sunlight energy, to convert CO2 and water into oxygen and glucose. Glucose then being used in cellular respiration to produce ATP. So again, photosynthesis is the process by which we convert CO2 plus water into sugars or glucose and oxygen gas with light energy. So photosynthesis, for those of you who don't know, photosynthesis is conducted inside the chloroplast, which is an organelle found inside plants, inside algae, inside cyanobacteria. They consist, and specifically, there are two stages of photosynthesis and two types of reaction within this. You have the light dependent reaction. You also have the light independent reaction, also known as the dark reaction or the Calvin cycle. And then with the light dependent reaction, that part will be responsible for the conversion of water into oxygen gas. And in the Calvin cycle, you convert CO2 and sugars. So in this case, glucose. Now, this process takes place inside the thylakoid, which is found inside the stroma, which is the space inside the chloroplast. And the thylakoid contains pigments such as chlorophyll. You have different types of pigments. You have chlorophyll A, chlorophyll B, and carotenoids that all play a role in absorbing light energy of different wavelengths. Again, this is just a preview of what this entails. So this is called a photosystem. These light harvesting complexes contain different pigment molecules that reflect light and transfer it to the reaction center, which contains a special type of pigment. In this case, in this picture, you'll have chlorophyll A, which transfer the electrons to a primary electron acceptor and sends it to the ETC, which stands for electron transfer chain, that allows it to create a hydrogen gradient and produce ADP and NADPH. So here you have a better example. You have photosystem 2. Light energy stimulates the light harvesting complex, and it's being reflected to the reaction center that contains these special chlorophyll pigments that then send the electrons to an electron acceptor. The electrons are then transferred through the membrane to these different electron transport chain, and then they go to photosystem 1 to produce NADPH. And in the process, you have these different hydrogens that are being produced here, here. And then so what's happening is that there's an increase of hydrogen inside the thylakoid interior, and this then goes down ATP synthase to produce ATP, which is the energy currency of all cells. And so, just to specify, the NADPH and the ATP is going to be used in the Calvin cycle, which is the process by which carbon dioxide is being fixated or fixed into usable sugars. So this is just a brief preview, a brief summary. So you have different wavelengths or different pigments. So these are different pigments that capture light's energy, passes it down to the electron transport chain, and then they produce ATP or NADPH. Water molecules split by sunlight to produce hydrogen and oxygen. And then also the light dependent reaction occurs inside the granule of the chloroplast where the thylakoids are found. So again, this is just inside the thylakoid. Now, the light independent reaction, which we mentioned briefly, takes place outside the thylakoid. So this takes place inside the stroma. It's the space inside the chloroplast, and you can just see that CO2 is being fixed into different intermediate molecules. There are different enzymes involved. So you could see that the ATP that's being produced here is utilized to convert this molecule into a sugar. And so in that process, you convert ATP to ADP and NADPH into NADP. And these again are being used in back into the light reaction and light dependent reaction to start this cycle all over again. Now, cellular respiration is slightly different. It's almost the reverse of photosynthesis. It's the process by which we use glucose and oxygen to produce CO2, which is what we breathe out every day, all the time. Water, which is through our urine, and ATP, which is in the form of heat, in the form of chemical energy or chemical reactions inside our cells. It's one of the reasons why we are considered warm-blooded creatures, because we use ATP to keep ourselves warm. So, and now we're talking about cellular respiration or aerobic respiration, which involves 4 steps. You have glycolysis, you have formation of acetyl-CoA, the citric acid cycle, and oxidative phosphorylation, which consists of the electrons transport chain and chemoosmosis. Again, this is just a brief diagram of what it looks like. You'll see that glycolysis takes place inside the cytosol. The cytosol is the liquid found inside the cytoplasm. It takes place outside of the mitochondria. And everything else, such as pyruvate oxidation, citric acid cycle, and oxidative phosphorylation, takes place inside the mitochondria. So glycolysis, at its most general explanation or brief explanation, it's the process in which we convert glucose or metabolize glucose into 2 pyruvate molecules. So that's an interest. The first stage of glycolysis requires that we input 2 ATP molecules to push the chemical reaction forward. And at the end, we get, because at some point we have 2 glyceraldehyde 3-phosphate groups, which 2 glyceraldehyde 3-phosphate molecules. And so the reaction happens twice. So at the end, we get 1, 2 times 2, that's going to be 4 ATP molecules in the second stage of glycolysis. But because we invested 2 to begin with, 2 ATP, so our net result is 2 ATP molecules. Okay. Now, the formation of acetyl-CoA is just simply pyruvate being metabolized by coenzyme A and NAD to produce acetyl-CoA. Acetyl-CoA then goes inside the Krebs cycle and interacts with oxidative acetate to undergo the different transformation of the different intermediates found inside that take place inside the citric acid cycle. And in the process, you'll notice that we're producing CO2. You also produce CO2 in the previous step. So CO2 is being released in the conversion of pyruvate to acetyl-CoA. And then you have, this happens again twice. You see 2 events of CO2 being produced, but because you have 2 acetyl-CoAs, you have 2 pyruvate molecules, then the total amount of CO2 is actually 6 molecules. Just keep in mind that in the process of glycolysis, you will end up with 2 pyruvate molecules. And so the amount of CO2 that's being produced, you have to multiply that by 2 because you have 2 molecules to begin with. Again, essentially the summary for this is that in each turn, you produce 1 ATP, 3 NADH, and 1 FADH2 in the Krebs cycle. Lastly, you have oxidative phosphorylation. This is the process by which we get all these. We have the NADH, which is the electron carrier, FADH2, which is also another electron carrier that transfers the electrons to these different protein complexes found inside the membrane of the mitochondria. And in that process, you're producing large amounts of hydrogen, which creates this proton gradient. And what ends up happening is that the proton gradient that's growing inside the intermembrane space, the protons will go down ATP synthase to produce ATP. So again, the main idea here is that the body has complicated but yet interesting ways in which it uses glucose and makes ATP, which is the energy currency of the cell, and plays a huge role in a variety of chemical reactions such as secondary messengers, gene expression, methylation, etc. Now let's talk about briefly how we could use this knowledge to help us with passing or doing well on the AP exam. So, for example, here's a question. So, a solution of bromothymol blue indicator is exposed to carbon dioxide until the solution turns yellow. An aquatic plant, Elodium, is placed into the yellow solution and exposed to sun. After a few hours, the solution is now blue. Which of the following statements best describe the purpose of this procedure? So, one of the things you have to consider is that this is called an indicator. So, it indicates something. They breathe in, they blew in carbon dioxide into the solution, which turns yellow. So yellow means it contains CO2. Okay. And then they add a plant exposed to the sun. After a few hours, the solution now turns blue. So it is a blue indicator. When they blow in CO2, it becomes yellow. It goes back to blue, so that means it no longer has carbon dioxide. So then, which of the following statements best describe the purpose of this procedure? This procedure proves that carbon dioxide is used up during photosynthesis. This procedure proves that CO2 is produced during photosynthesis. Okay, this procedure proves that carbon dioxide is produced during photosynthesis. Well, we know that's not true because carbon dioxide is not produced during photosynthesis. So by the process of elimination, we get rid of that one. The procedure proves that bromothymol blue as indicator becomes bromothymol when exposed to carbon. We have no indication, no evidence to believe that. There's nothing that tells us that. You're not supposed to, we don't aren't supposed to know the full chemical processes of these indicators. So that's something we could rule out. And then it says the procedure proves that carbon dioxide is used up during photosynthesis. The procedure proves that oxygen is produced. Even though B is true, that procedure proves that oxygen is produced during photosynthesis, it's not related to the indicator because it's talking about carbon dioxide. So the procedure proves that carbon dioxide is used up during photosynthesis. Okay, so that's how we choose that answer. So the process of elimination. Question 2, carbon exists in multiple isotopes with similar chemical properties. Carbon labeled in this labeled is used. Carbon labeling is used in chemical and biological experiments, which involves replacing regular carbon atoms with radioactive carbon-14 atoms that can be traced in chemical reaction. A group of plant samples is grown for several days in an environment containing CO2, containing radioactive carbon-14 instead of non-radioactive carbon CO2. Which of the following statements describes the most likely result of this experiment? So let's talk. So Rubisco carbolase will display increased activity in the plant cell while they undergo photosynthesis. There's nothing that indicates that, so we can rule that out. Then the next one is radioactive water will be released from the plants. Well, carbon dioxide doesn't really play a role in the formation of water, so no, not that. Radioactive carbon-14 will be found in the starch stored in the roots of the plants. Radioactive carbon-14 will be found in the leaves of the cell. So although the leaves are made of cellulose, which contains carbon, it has nothing to do with the fact that with the production of the starch, which is a carbon source, which is an energy source. So in reality, because we remove 3 options, the only option is C. So again, via the process of elimination, we're able to find the answer. Now, let's go with the third one. A researcher adds carbonated water to a potted plant every day and notices that the plant is bigger and greener than other plants in the same location. Which of the following best explains why the carbonated water affected these results? So let's see, the carbonated water is alkaline, which causes rapid growth in plants. That's not true because carbonated water is actually acidic, creating carboxylic acid. So it's actually going to be acidic, not alkalinic. So that's wrong. The roots of plants absorb the oxygen from the carbonated water, so that's not true because plants absorb they absorb CO2, not water, not oxygen. So that's not true. So we already eliminated 2. The roots of the plants absorb the CO2 from the carbonated water. That seems to be interesting. The leaves of the plants are exposed to more CO2 and water from the carbonated water, both of which are required for photosynthesis. Well, that's not completely true. The roots of the plants absorb CO2 from the carbonated water. So it seems as though the best option would be C, not D, because the leaves of the plants are exposed to more CO2. That's not true because we're talking about the roots of the plants because you're adding carbonated water to the roots. So that's not true. So because of that, it's not true. So again, via the process of elimination, just knowing the basic elements of plant biology will help you do well on the exam. Lastly, A botanist performs an experiment to explore where trees gain the raw material needed to increase in mass. She plants a seedling weighing 3 pounds in a garden box containing 150 pounds of soil and regularly waters the seedling. After 3 years, the botanist weighs the tree and soil. The tree weighs 32 pounds, 132 pounds, 5 ounces. The soil weighs 147 pounds, 10 ounces. Which of the following statement best explains these differences based on her data? So we know that she added in the beginning 150 pounds of soil. She's a seedling that weighs 3 pounds. So essentially, from 150 pounds to 147 pounds, that's actually nothing. So then the question becomes, where does the plant get the mass from? So let's see what are the options. So the seedling mass increased by incorporating soil from the garden box. Well, we know that's not true because you don't get 147 pounds from just a 3 pound difference. So we know that's not true. The seedling mass increases by incorporating carbon dioxide from CO2 in the atmosphere. That seems a possibility. Okay, let's see the next option. And so the seedlings mass increased by incorporating water from the botanist regularly watering. No, because water is only being used as carbon dioxide as oxygen. So it actually every time it uses water, it will release it in the form of oxygen. So that's not true. And the seedling mass increased by incorporating water from the botanist regularly watering and soil from the garden box. So it's not true because we know that the soil it's only 3 pound difference. So we know that's not the case. So we know it's going to be B. Again, via the process of elimination, we know that that's the answer, and that's the one of the better strategies and the best strategies to do well on the exam. By the process of elimination, eliminating the things that you know for sure are not going to be the answer will increase your chances of getting the right answer at the end. Well, that's it for today. Thank you for joining me, and I look forward to seeing you next week. Take care. Bye bye.